Welcome back, everyone. Critical test. Can you hear me? I think you can hear me. I can unmute myself this time. There was no confusion about it, but I'm going to ask for some confirmation in the chat that you can hear what I'm saying. Yes, thank you, Andrea. Excellent. So, okay, good. So there's a few of us still here. Let's think back to last year. It was around this time we did our first ever summer school, and it was 5 days. We wondered if that would be too much. Now it's 8 days, and you're on day 7, and you're toward the end. And we've expanded the material, and we've gone even deeper into some really interesting stuff. And it's an increased breadth and it's an increased depth. So congratulations for getting this far. And we're not done yet. There's a lot of really great stuff to come, but it's occurred to all of us. I mean, it's been a year of this, and you're all pretty tired of sitting and listening to videos on your computer. We wish we could have done it in person, and hopefully we can do that next year. But we've been sitting now, at least I know I have, a lot of us have been sitting for hours, and that can increase your risk for deep vein thrombosis and blood clots in your lungs, and we want to avoid that. So just, let's just take a second. I know you'll do this. So this last session is going to be a breeze. Don't worry about it. There's not a lot of math. I don't even know if there's any math. It's going to be a bit shorter. So just get up out of your chair for a second. You don't have to, do anything. You don't have to put on a workout outfit or anything. Just stand up. Just shake it out, Do some running in place. Just for a little bit. Get the legs moving. Okay? Just shake it up. You gotta get the blood moving in case there are any deposits on those veins. Just, it helps the brain. Maybe some jumping jacks. Just a couple. Just whatever it is. Just stand up, Maybe you probably didn't even stand up during the break. We sit there and we're on our devices. Stand up. Jumping jacks. And just, get moving. We used to do planks. So if you come work in the center, we'd sit and we'd do like planks for core strength sometimes. It's just good to interrupt the workflow. So a little bit of movement, get the brain going. I swear we've got like an hour left today. We can chat, we can think about these concepts. You feel better if you just stand up, move around. I think it's a nice little way to break things up. So, that said, you can keep working out, keep moving while I talk. I mean, I think most people are probably multitasking anyway, so stand up, keep moving while I'm talking, and just, you can have the slides on. I won't be offended. I can't see you not paying attention, so don't worry about it. But this is going to be really interesting. So I'm going to share my screen here. It's a testament to my deteriorating cardiovascular fitness that felt pretty strenuous. So jump up and down for a few seconds. Okay. But you got to start somewhere, right? What do we say about starting habits and improving our fitness? Are we going to do it one day, or is today day one? How many of you have seen that on Instagram? Probably everybody. Okay, so it's good. We feel reinvigorated. So here we are, session 4 of day 7. We've heard today some really interesting stuff about Abhi's work, an area of expertise in digital health, and I mean, that really is, it's probably the next major frontier. It's probably the biggest, one of the most talked about, at least, kind of innovations and directions for, capturing data and getting past a lot of these barriers that we're bumping up against in the study of psychiatric illness and just in mental health in general, is because we recognize that in contrast to something like cancer, where the tumor kind of grows but doesn't evolve and change on a minute-to-minute basis, your mood and your experiences and your behaviors do. They change incredibly quickly. You need that kind of high high density data in the real world, throughout your life in order to kind of get past it. So really important, right where we're kind of going to. I'm going to talk a bit more, I'm going to back up a bit and talk about precision medicine, which is a word we've heard a lot about over the last 7 days. It's a word you're probably familiar with, 2 words, a term. And I just want to give you some definitions, some perspectives, some examples, and a bit of some state of the art, a couple examples of studies of precision medicine in psychiatry that involve data types that we've looked at. And then I'm going to do a little bit of a tease for tomorrow morning so that you still feel motivated to get up and attend tomorrow's session at 9 a.m. through to lunch. And that will kind of be the extension of this session tomorrow morning. And then we'll have our lunch, and we're going to bring everyone back in, everyone from all the previous days, and do an afternoon of kind of open discussion with us. And, bring your friends, bring your family. If you have anybody who wants to just talk about any of the concepts you've heard about, even in passing, we can start a conversation about that. If you have a research project and you've got a really specific question about a methodology, we can answer that too. That's going to be the idea, kind of wrapping it up with tomorrow. So here we go. Today I'm going to talk a bit about precision medicine and a bit of introduction to interactive methods and integration in psychiatry. It won't be the full time. I'll try to keep it succinct. So, again, what is precision medicine? We talk about this a lot. I think most of you will have a pretty good idea of what it is. I'll start with kind of when it is or when it was. It's been in the lexicon for a little while, but it hasn't hit mainstream. Kind of the idea of precision medicine has been a goal of a number of fields for decades. For psychiatry, a bit kind of in the mid-thousands more so, it kind of entered, precision psychiatry became a thing. But precision medicine has been around for a while. I'll talk a bit about that. 2016 was a big year because President Obama said, okay, precision medicine is really important, and this isn't specific to psychiatry. Precision medicine is important, so we're going to invest $250 million roughly over the next many years, kind of put it in the 2016 budget to establish infrastructure to support research that could basically produce precision medicine across disciplines. And the whole idea of precision medicine is kind of arises from the lack of precision medicine. We've heard about this, I think, in 3 or 4 different talks. It's just the fact that regardless of your affliction, whether it be cancer or major depression or diabetes, no drugs work for everyone. There's just the chance that your pharmacological intervention won't be effective for you. And there are reasons why. When you go to see the doctor, and we heard about this on day 2, you're not seeing a robot, right? You're asked questions about your lifestyle. They know things about your age, about your biological sex, about other aspects of your lifestyle. They make decisions. There is some tailoring of medication to you, right? It's not completely blind, but yet what this is saying is, despite that tailoring, despite our best knowledge and the most kind of the most we can possibly, or any clinician can integrate their knowledge on medicine about these drugs and use available guidelines, there still is this just chasm between what we hope will be effective and what is effective. So the kind of one of the formal definitions provided for precision medicine, at least of the concept, is one in which healthcare is individually tailored on the basis of a person's genes, lifestyle, and environment. And again, this is not new. So people talk about it like it's newfangled. It's not a new thing. You can think about examples from other fields of medicine, like blood transfusions. You can't just give any blood to any person. You have to know what blood type they are to match the blood sample. So this is where there's this taxonomy for precision and personalized medicine, and this figure can maybe help you get around the terminology in case you encounter it in your training and in your conversations with other people. Stratified medicine, precision medicine, P4 medicine, like predictive, preventative, personalized, participatory, and personalized medicine. The fact is, they're all encompassed. This is not a precise figure. This doesn't really do it for me. They have some overlaps and not kind of overlaps. One of the points that I want to make that helps me think about this, and really is the only true distinction. Oops, if I click over here, I lose the... There we go. Is a matter of... oddly, a matter of precision. You can think of it as granularity or resolution. So when you talk about precision medicine, stratified medicine, P4 medicine, it's the idea of finding groups of people, broad groups of people, like blood types, for which, you you might prescribe one type of medication, a broad type, or another type, is acetaminophen here, ibuprofen there, or something like that, 50-50. Whereas personalized medicine, individualized care, everything else, it is kind of what it says it is. It sounds self-explanatory, but the terms aren't completely interchangeable. As soon as you start talking about personalized medicine, some people get very touchy because they say that's impossible. Why are you even trying to do personalized medicine? Basically what you're saying is everybody's an N of 1. Every study is an N of 1. Every treatment should be completely tailored to one individual and driven by science. That idea mostly came from genomics, right? The kind of idea of a fingerprint, a signature per person. And there's, so far no real, examples of that, the kind of success from a statistical study standpoint of individualizing, each person. It's always grouping, it's always stratified. And so that's exactly what this figure is. We've seen versions of that. Again, on the left is one drug for everybody, and some people benefit. On the left, diagnostic biomarkers. Therapy is tailored for the groups of individuals, some larger groups, some smaller groups. And that's like the future. The future is already here in oncology, to some degree also in immunology, treatment of HIV, and metabolic disorders. There is some there's some applications in mental illness, which I'll show, but it's key to look at the numbers here. So the definition here of a drug that counts as something that is a precision medicine or precision medical approach is just a drug that point to a specific biomarker in their label to guide use specifically. So there may be applications of some medications where the clinician is taking into account extra information, but that would be not guided by empirical peer-reviewed knowledge or embedded in guidelines for the actual use of the drug. And in the United States, there's about 286 of those medicines that would be defined in this way on the market, virtually all of them for cancer. And you can think about, we can think about why that is. You can ask me why that is. I don't have, some ideas, but it has to do with complex interplay between funding and societal perspectives and complexity of the disease. In psychiatry, there's been some success from this field of pharmacogenetics. So again, a lot of the foundations of the idea of precision medicine came from the sequencing of the human genome, or just kind of learning about genetic structure and genetic risk for illnesses. And also discoveries of kind of key roles of specific genes in regulating really important generic processes. So the idea behind pharmacogenetics is that there are these aspects to how drugs work in your system, these pharmacokinetic and pharmacodynamic aspects to a medicine's journey through your body that are really in humans and other organisms directly influenced by some specific set of molecules. So pharmacokinetics, think about how drugs are metabolized, essentially, how it goes from a pill or an injection into your system, how it's processed by the liver, how it enters, the circulation through your stomach or through the intestine, and exactly how it's broken down or how it's processed. Pharmacodynamics would be, how does this drug act in your system at a functional level, right? How does it bind to the target receptors? How specific is it to the target receptor? Does it have other interactions, and is it broken down into other metabolites that themselves may be active and having certain dynamic properties? So, does it elicit an immune response? These are kind of two classes that can both be acted upon. This is an example of a test. You can see on the left-hand side, there's a set of 6 genotypes. These aren't genotypes per se, these are genes for which there are genotypes. Each of these genes, at least the ones on the top here, these CYP genes are cytochrome P450 oxidase genes. And you can see I've highlighted the liver here because I'm trying to highlight that a lot of the early success in pharmacogenetics applied to psychiatry is the fact that a lot of antipsychotic drugs a lot of drugs of other classes as well, but it turns out that antipsychotic drugs in particular can be metabolized, or they are metabolized by these enzymes in the liver. And depending on your genotype at certain specific locations, you may have a very high rate of metabolism or a very low rate of metabolism or something in between with varying allele frequencies. So it's actually quite an informative thing to know about someone. Dosage is really important to know, not just which drug, but how much of a drug. If you give too much of a drug, it might have side effects. If you give too little, it might not be effective at all. So dosage and titration of a dose is really important. But if you are a really hyper-fast metabolizer of a particular drug and you break it down very quickly, quite often the way it works is the drug is processed in the liver and becomes active. It's an active metabolite of the drug. Then maybe if you have a really high metabolizer status, you need a very little amount of the drug to be for the drug to be effective. On the other hand, if you have a very slow metabolism, a lot of that drug may pass through your system inactive, and you might need a lot more of it to see the result, right? And it may be different for different medications. So really like a very concrete mechanism to kind of act on. And then there are others, kind of transporter activities, as well as receptor activities for serotonin, 5-HTR2A. Those might affect, if you have certain genotypes, they change the shape of the protein, the shape of the receptor, and they change its function and its response to the same medication. So there are companies, and CAMH partners with the Myriad Genomics, I believe now they're called, and this gene site test, which I'll show you, where you have this, you can see this interpretive drug sorting, effective composite genotype on each drug. This is polygenic scoring. It's a little more complex than the algorithm I showed, but it's a proprietary algorithm that companies have to calculate what the combined effect of all of these genotypes are for an individual person, and then group that person, again, precision, not personalized, into one of three groups for each medication. So you have medication A. According to all your genotypes, you should use this medication as directed, or you should use it with caution, or you should use it with caution and more frequent monitoring. Of course, no one's telling the doctor exactly what to do, but basically saying, look, this person has a rare genotype, they're going to metabolize this medication really fast in the liver, it's going to pump out into their brain, it's going to bind to their receptors that are hyper-receptive and hyperactive, and give them a small dose of this drug. If you give them regular dosage, if you use the medication as directed, they might have really serious side effects or an increased risk of side effects if you just kind of go with it. And there's some success there. This is an example of an actual test that you could get, GeneSight. Don't think it's currently paid for by OHIP. It's an ongoing battle with these things. So Jim Kennedy at CAMH has partnerships with this company, Myriad Genomics, to help develop GeneSight. Of course, these algorithms are based on things like GWAS, genetic association studies, to understand how you calculate someone's score using genotype data. Just like we saw in day 3 when I was talking about polygenic risk scores, how do you know what weights you give to the CYP4 P450 oxidase genes, right, or the serotonin receptor? You know, how do you choose what puts this person into one of these categories when it comes to particular medications? So there's a lot of research that's necessary. This is where we're at, and it's a battle to convince regulatory agencies that the benefit of doing the genotyping, running it through the proprietary algorithm, which costs money, oddly enough, these are not open source, as well as delivering this test to your physician. This could be your family doctor reading this, or your psychiatrist more likely. Because I don't know if they give it to first-line physicians for this test. But whoever it is, it's someone who's probably not used to reading a test like this. Psychiatrists aren't used to and often don't believe in genetic evidence, this kind of type of precision medicine to guide their care. So it's new. It takes adoption. It takes the government believing that it's worthwhile. Early research suggests that it is effective, but it's very difficult to study how effective something like this can be, because you need many examples of people who are enrolled in research studies who are currently taking who are currently taking a medication that they shouldn't be on according to their genetic profile, and then have them switch to one that the genetics say they should be on. And then you can gauge whether or not that change is a good one, right? You have to have enough discordance in your sample. A lot of times these tests mostly look like you have a lot of drugs in the green category. It's usually the rarer genetic variants or the more uncommon ones that produce extreme phenotypes for the pharmacogenetics or pharmacodynamics and pharmacokinetics of these drugs. So But this is real. It's happening today. You could buy this if you live in the United States. You could just say, hey, I want a genetic test for my treatment, and I want it to be directed by genetics. As I said, these decisions are more traditionally based on just a few genetic variants. I talked about polygenic risk scores, where you take the whole genome, get really, supposedly better, more information. It's better from the perspective of capturing more heritability of the trait, but when it comes to calculating a score and telling you if you're at risk for something, this plot kind of sums up the reality of the situation when you're looking at polygenic risk scores, and that is they are pretty much normally distributed in the population, in the general population, people who are sick and people who are healthy. And the people who are diseased, if you separate them out, the distribution kind of shifts a little bit. On average, people with the diagnosis will have a higher polygenic risk score than those without. I mean, that's the whole point. But if you pick one person and just plot them on this genetic risk score, unless they're really in the very, very top percentile of risk or the very, very low percentile of risk, it's very, very difficult to tell anybody with any certainty whether or not they're going to be healthy or they're going to develop a diagnosis in terms of risk. And the same applies to treatment, right? It's just a phenotype. Are you going to respond to a treatment or not? We can generate a polygenic risk profile or a general risk prediction score, and this type of predictive ability, comes down to just kind of odds ratios and positive predictive value for a test. There isn't a lot of confidence with which we can actually go to one person and say, you are really likely to develop a disease. And there's a lot of research going into this, trying to better the kind of foundational pharmacodynamic and pharmacokinetic findings that have come from mostly, quite largely candidate gene studies from the last 20 years to build on all of these huge resources and genetic studies that have been built. And there are some interesting patterns that come out of this. So basically the idea is we have all of these GWAS for psychiatric illnesses. There are even some GWAS for response rate, although those are, I think, the largest ones in like a few hundred people, 300 or 400. GWAS on a few hundred people is not powered adequately and not expected to replicate. So you kind of might expect something like this. You develop a polygenic risk score for the severity of an illness like major depression. And then you go into a clinical sample of individuals who've been diagnosed with major depression, and you give half of them treatment for first-line treatment. Maybe it's a selective serotonin reuptake inhibitor, and maybe the severity of genetic risk will dictate the degree to which they respond to medication. And something else kind of happens, or has been happening. Number one, as I mentioned earlier, the effect of these polygenic risk scores in terms of being able to differentiate between even just the diagnosis itself that is kind of targeted to split is not very high. So in this kind of real, this is a study done in actual health care, 4 different healthcare systems in the United States, 106,000 patients, calculating polygenic risk scores for schizophrenia, and look to see, okay, how many people with schizophrenia have a high risk score, how many people without schizophrenia have a low risk score. It's basically a discriminant analysis between these two. The odds ratio, right, for this score is 1.55, and this is an odds ratio where you take the people who have the top 10% of scores, the top decile of scores. So immediately you're only looking at 10% of the sample, and the people who have the bottom 10%, so the lowest possible score, and then comparing in those top and bottom deciles what proportion of people do and don't have schizophrenia. And the ratio of the odds, right, for being schizophrenia versus not in those two groups is only 1.5. Like, your odds of having schizophrenia are only 1.5 times bigger if you're in the top 10 percentile of scores versus bottom 10 percentile. And remember, that's 80% of this 100,000 person sample in between. It's not a very clinically useful metric, and they're often criticized for that. Not all findings are this unremarkable, but I'll talk about clinical utility in a bit. One thing that I really want to point out here, and feeds into this question of how polygenic scores are used for treatment, and one of the surprising patterns that's emerged, you'll notice that the schizophrenia risk score is most effective at differentiating between schizophrenia cases and controls. I should actually say this odds ratio of 1.5 is for the top 10% versus remaining 90%. So it's not quite as dramatic. The green circle is top 10 versus bottom 10, but still it's a little over 3, odds ratio of 3. But look at the confidence interval; it could be as low as around 2, and so made it a bit more dramatic. But it's still not it's not fantastic. It's still top 10 versus remaining. Remember, it's not. It's not top 50 or bottom 50, and it's not a continuous feature. It's not a predictive value like an area under the curve. But what I want you to notice is schizophrenia is the most, and then psychosis, and then schizophrenia. That's fine. But then you have post-traumatic stress disorder, bipolar disorder, suicidal ideation, substance addiction, agoraphobia, social phobia, panic disorder, anxiety, viral hepatitis C, personality disorders, viral hepatitis. These are all diagnoses in this patient population that were statistically significantly predicted by a polygenic risk score for schizophrenia. So in the title, they're talking about penetrance, which refers to the proportion of individuals in a population that exhibit the phenotype that the genotype is a risk for. And then pleiotropy, which is the idea that a single genetic causal factor, a single genetic variant in this case, or set of variants for one disease, this polygenic risk score, influences many different traits. And that's the case. So notice this study is a polygenic risk score for major depression is associated with response to lithium treatment in bipolar disorder patients. Bipolar disorder is not major depression, as we learned on day 2. Very different clinical presentations, in fact, different genetic bases. And this study did test a polygenic score for bipolar disorder. They tested a number of different polygenic scores. It was the polygenic score for depression that predicted response to lithium in bipolar patients. And notice the odds ratios, which dance around. The plot looks kind of fun because of all the curves. You'll notice the polygenic score decile. So as I mentioned, quite often what's done is you split the polygenic score distribution into some type of percentile, deciles, quantiles, tertiles, because you need a way of getting a kind of practical number, a way to stratify. Again, it all comes down to precision medicine, group stratified medicine. Who's high risk, who's low risk. It's a very kind of clinically useful or interpretable way of going about it. So depending on how you split the deciles and compare to the remaining sample, you can get an odds ratio of up to a little less than 2, and it varies depending on the ethnicity of the sample. They had a multi-ethnic sample. The methodology in this paper is somewhat flawed. They're looking at variance explained. Notice how it's 0.7% of variance. It's not 0.7 r squared. It's 0.7% of variation is explained by the continuous version of this score. Very small effect sizes in isolation. Again, here's a result for a polygenic risk score for major depression associated with change in depression scores from baseline. Meta-analytically kind of significant, but this was not statistically rigorous in this paper. This was also a polygenic score for neuroticism, which showed the same downward effect. These are neuroticism and major depression are overlapping to some degree, but what is emerging, and I'll continue to show, is that these scores are not predictably associated with what you might think they're associated with. Here's another example. Higher polygenic risk score, again for schizophrenia, may be suggestive of treatment non-response in major depressive disorder. So this isn't lithium in bipolar patients, this is major depressive disorder. And this has to do with some features, and this is something that we heard about a little bit on day 2, again, which is kind of possible comorbidity or underlying burden, like a genetic burden, risk burden for psychosis or for kind of more severe psychotic symptoms may be related to how individuals with major depressive disorder and as well bipolar disorder and other disorders respond to their treatment, or at least kind of experience their trajectory in the healthcare system. But again, in this case, you've got a polygenic risk score for schizophrenia. Methods were not rigorous. The p-values were not quite significant. This is a p equals 0.02, and again, they're separating into quantiles. So if you take the 5th quantile, I actually don't remember how they split this, but this is the highest resolution. So it's just like if you take the top, I don't know, maybe it was the top 20%, if that's how they split it by 5, or if it was just the top quantile. It doesn't matter, because I was underwhelmed by this study. About 1,000 patients with major depressive disorder. The underlying, the result is that the major depressive disorder PRS was not informative whatsoever. They checked other scores as well. And this result was not striking either, to be honest. So it's kind of disappointing, all around disappointing. You'll read a lot of criticisms on polygenic risk scores and use for clinical utility, but it's where we are right now. It's kind of where things are sitting. There are efforts to go beyond go beyond kind of more basic polygenic scores. We talked about some different polygenic scoring algorithms, and some of these studies I mentioned use different types. They aren't all using clumping and thresholding. Some of them are using Bayesian stats. Some of them are using, LD-pred as some kind of machine learning to select SNPs. There's also this funny review. It's artificial intelligence and machine learning approaches. I'll draw your attention to Chang et al., this 29th citation, who used linear regression. What you choose to call artificial intelligence and machine learning is really it's really up to you. You just got to decide how much of that language you want to use, but you can really get away with it all. You'll notice most of this is kind of random forest, tree-based, decision tree-based ensembles. Those are they're nice because they're fairly interpretable. We're going to talk about tree-based methods tomorrow. I'm going to talk about XGBoost and SHAP a little bit. The idea of this is to kind of say that people are working on using SNP data and GWAS data in various ways to get at precision psychiatry, to make better predictions of who's going to respond to what. The accuracies vary enormously, 52% based on SNPs. Some of these are not all SNP-based. You have to see clinical and demographic data sets, area under the curve of 0.72, it's a correct classification. There's a lot of other... they're all based on some genetic data, but this is kind of, some people are adding in more stuff. 0.92 for lithium therapy responses. There's also a really interesting finding where in machine learning in general in psychiatry, the higher the reported accuracy of your algorithm, the smaller the sample size of the study, and that's not a mistake. That's bias at work. So the answer, I can't tell you exactly what the answer is. It's highly likely that the answer is that, of course, SNPs on their own are not really that informative. A lot of people are looking beyond SNPs to rare variants, and not just rare variants, but structural variation, other types of genetic variation, epigenetic modifications. What I'm saying is it's integration. So far we've focused on a fairly reductionist perspective, which has paid off a lot. Looking at genetic variants for CYP genes in the liver has been incredibly useful. They've already developed clinical tests based on that, and this is work that came out in the early thousands. But it's like low-hanging fruit, right? So it seems that in order to kind of build more reliable models that do replicate across domains and across populations, and have some generalizable clinical utility, there is more to look at, and there's more integration. You have to look at more data, and that's a lot of what I'm going to be talking about tomorrow. We've got a question from Akshay. Based on the slide you were showing that showed statistical significance of polygenic risk scores, will it be correct to say that psychosis or psychosis-like symptoms are more likely to be predicted than other mental illnesses? So no, I didn't want to make that. I didn't mean for that to be the message there. So this was particular studies where we're looking at treatment efficacy for major depression or kind of lithium treatment. I wasn't trying to draw comparisons between how well polygenic scores for different disorders fare in out-of-sample prediction, but there are a lot of examples of that. I think tomorrow I'm going to present a study that shows exactly that. That's not specific to psychiatry. It does compare major depression to a lot of other disorders. But some diseases do fare better in out-of-sample prediction than others. There has been no really comprehensive study looking across methods, across diseases, and in multiple data sets in psychiatry and psychiatric illness to evaluate how well and kind of put a real number, like a quantifiable risk number, on how well different diseases perform. Yeah. From our own heritability of psychosis higher than MDD. I don't actually know if it is. I think maybe it is. I had a plot. I mean, I haven't memorized the numbers. I had a plot earlier in my third slide. There are tons of meta-analyses for heritability of psychiatric disorders. It's not hard to find those numbers. I'm not sure how much more or less heritable it is. Yeah, in theory, SNP-based heritability should help inform how well a polygenic risk score is at predicting the illness, but it's not just an estimate of SNP-based heritability, it's also phenotypic heritability and heterogeneity between data sets. Because sometimes you have within sample predictions, I mean, they're internal validated R-squareds and internally validated predictive algorithms, but as soon as you jump to another population, another data set, it disappears. The effect is, I mean, doesn't completely disappear, diminishes really enormously. Yeah, we've actually been working with that quite a lot. It's a very active field of study. So moving on, it's not just genetics, and I'm not the expert in this, but I'll speak a little bit to some interesting findings that have come out. One paper developed a machine learning method, they call it CELSR, which uses some inputs from EEG, a resting state EEG, for individuals who are prescribed sertraline, which is it's an SSRI in major depression. And these patients who receive treatment, actually some also received placebo, and there is a scale called the HAM-D, which is a measurement of depressive symptoms, and the measure that they used was a change in HAM-D over time. So you receive a treatment for a disease, you receive a pharmacological treatment, and check to see if you get better or not. And they train this model, it's a machine learning model that uses fairly canonical bands, wavelengths from, again, as I said, the resting state EEG. So it's, John can speak to this paper perhaps in more detail in terms of what they did. My understanding is that they took fairly vanilla metrics that come from this resting state MRI, but used some deep learning algorithm to extract signals, some black box signals essentially, which they've mapped here onto the cortex. You can kind of see where these latent patterns of signaling are coming from to best fit a model. Again, using machine learning, so this is iterative, right? They fit a model, they have a cost function, they test to kind of optimize that model without overfitting. What they observed is that they could predict. They could have these predicted change in HAM-D scores. This is in Figure A for the sertraline group that actually predicted HAM-D change in real life. So they used this EEG information only to predict a change in HAM-D, and that aligned, correlated with the actual change in HAM-D that people had. And that happens when they trained in the people who were treated and tested their algorithm in the people who were treated. They claim that when they test their algorithm in people who received placebo, there was no prediction here, which is so what they say is that this effect is specific to the pharmacological intervention because of its lack of predictability in the placebo group. They then went on to a secondary sample. It was a sample of 72 people from an independent group who they grouped into responders and non-responders to treatment, and then they predicted the HAM-D scores and looked to see if their predicted HAM-D change scores from the CELSR algorithm was different between the partial responders and the ones who were resistant to treatment, who didn't respond. And there was a difference. They found that the predicted change was lower in those that didn't respond. So it sounded nice. This paper received a lot of attention. It got a lot of flack. People were very upset about the inference from panels A and B, whereby training your data set in this kind of sertraline exposed group and testing within that group using a cross-validation framework, they fit this model. So essentially, the data that was used in validating this model in a roundabout way was also used in testing it. What they found here is that when you train it in the sertraline group and then test in the truly external placebo group, there was no predictive value, but there's another explanation, which is not that the mechanism is specific to the pharmacological group, but that it was actually just overfit in their original data set and did not generalize well to unseen data. And then in this case, in the second half, they said that there was this fallacy, this transposed conditional fallacy, which is in this analysis, they inverted their predictive algorithm. So in their initial analysis, they predicted real change in HAM-D with predicted change. There's a lot of predicted words here. They used their algorithm to predict real change, and that was how they, kind of validated their model. And then in this second sample, they didn't actually do the same thing. They looked to see if they could predict their predicted score as a function of partial responder or treatment resistant. So in one case you have the probability of something occurring, the probability of a true change in HAM-D given the predicted change, and then in this case it's actually the other way around. It's actually the probability of the true change occurring, or it's the probability of this HAM-D prediction that they're having from their algorithm given partial response and treatment response. And Frank Harrell, who does the RMS-R package, reanalyzed their data and found that if you actually do the exact same prediction, if you take the resting state data from this out-of-sample analysis and just try to predict, as they did in their original analysis, the true change in HAM-D over time, the AUC, the differentiation, is very poor. It's just over chance. It's like 0.55 or something. But the fact that it received some, is kind of controversial somewhat, does not completely invalidate it. People butting heads. You very rarely see editorials in Nature and commentaries and Matters, scientific Matters articles coming up for interesting papers like this that aren't published in Nature Biotechnology. So it's a ripe field of investigation. Another thing I wanted to point out is beyond, there's genetics, there's some EEG, there's a whole field of fMRI, functional MRI, biomarkers, and structural MRI biomarkers as well that you should be aware of. That's, again, not my primary area of expertise. One thing that I find to be really striking, which is interesting, is fluid biomarkers. This is interesting because it's very it's not an intrusive method of testing like genetic testing. It's a blood test. So we can take a blood test and measure inflammatory cytokines and molecules in the blood. So these are signaling molecules, interleukin 2, interleukin 4, interleukin 5, interleukin 10, and IFN gamma, this is interferon gamma, tumor necrosis factor, various molecules that are used by the immune system for signaling to other cells in the immune system and other peripheral tissues that there is something wrong, that there needs to be an immune response, or that the immune response should be kind of abated or deadened. And in this sample, it was a small sample. They had 71 people who responded to treatment for major depression, and 33 people who were not responding to major depression. What they found is that between baseline and 12 weeks, they did blood tests at both, the levels of anti-inflammatory cytokines increased universally. Regardless of whether or not you responded to treatment, almost universally, the levels increased. And yet the pro-inflammatory cytokines changed in a way that was unique to the people who responded. Right? Pro-inflammatory cytokines also went up in the individuals who did not respond to treatment, but in those who did respond, the pro-inflammatory signature declined over those 12 weeks, went down from baseline. So it's a really neat kind of molecular signature of specific pro-inflammatory factors that might mark that that's kind of successful change. Unfortunately, at baseline they also had higher levels. So this is the pattern is there, but the predictive ability to measure this at baseline is not quite there, although this is a really active field looking at inflammation and inflammatory states and how they might relate to depressive symptom severity and your likelihood to respond to treatment. And it's been studied for a while. Inflammation is something that occurs as a result of a lot of things, a result of stress, a result of hospitalization, a result of infection, and it's been noticed for quite some time, certainly in geriatric psychiatry, and that's the area in which I work a lot. When you have traumatic events, stressful events, inflammatory conditions, even things like arthritis, you can have very dramatic shifts in someone's psychiatric state, someone's mood. In midlife and even early life for major depression, you see patterns that are pretty robust at the meta-analytical level. So this is a meta-analysis recently published looking at a number of different anti-inflammatory agents, including non-steroidal anti-inflammatory drugs like ibuprofen and kind of common over-the-counter anti-inflammatories, but also other like different classes of what could be considered anti-inflammatories. Trials of DHA and EPA, so these are lipids, right, fatty acids, these long-chain fatty acids, and all pretty much across the board. I mean, at least in the meta-analytical state, the prescription, the taking of anti-inflammatory agents either on their own or as an adjunct during treatment is significantly associated with responses to medication as well as to severity of major depression. There are several reviews like this. The effect sizes aren't dramatic, but it seems to show this kind of really nice evidence that anti-inflammatory drugs and that mechanism of inflammation may be something really worth targeting. It's also something that we can see with brain imaging modalities in some cases, and also something that we can get at in terms of looking at pharmacogenetics and the genetic regulation of inflammatory factors, pro-inflammatory and anti-inflammatory. So, promise. But cautionary tales for all of it. Recognizing diversity. We've heard some of this, so I'm not going to dwell on it. I talked about the lacking diversity, especially in genomics research, and we've seen various versions of this plot, which shows the distribution of different ancestries in available genetic resources that have gone into GWAS. So if you're calculating a polygenic score, if you're interested in doing a study that looks at major depression risk, or even in some cases response specifically for precision medicine research, your algorithms will not be able to generalize very well beyond European ancestry. I did point this out the other day. I added this figure in because I linked to the publication that talks about this, and just one of the ways in which having various populations actually improves your ability to detect signal, true biological risk signal, causal signals in GWAS, and that's because you're including tag SNPs for haplotype blocks that are unique to individual populations, but kind of incrementally, by including a whole bunch of them, boost the overall power of that GWAS. Because if it is a truly causative variant, then that power kind of increases. There are also cases of different causative variants in different populations, and I mean, to be honest, I'm not sure exactly how they model that, but I wanted to put this reference data up there. The reference there is on the bottom, although it occurs to me you might not be able to see it because I think they use Tractor for this analysis. You might be able, okay, I can't move my face, but I'll make the slides available so that you can see the citation for the paper. I also think I posted it in Slack, or at least I posted a relevant one. But this is Alicia Martin's work, just showing the implication of that, specific implication of that for precision medicine. So this was across 17 different quantitative traits. This was not specific to psychiatry, but the picture is never better in psychiatry, I assure you that. The accuracy of the predictive algorithms, so this is kind of like brought up a bit, actually I mentioned, is the predictive accuracy different for schizophrenia, better for schizophrenia, worse for major depression, better for, autism spectrum disorders. It's certainly much worse if you're not a European, if you don't have European ancestry. And the prediction can be really bad. So if you take for these 17 diseases, the predictive accuracy as a benchmark of 1 for the European populations, you're at less than 25% of that prediction accuracy if you have African ancestry. So the paper is basically entitled, using polygenic scores that we have today for clinical decision making will do nothing but exacerbate the existing discrepancies in our healthcare system and really magnify them. And that's that's where things are currently at. If you're interested in studying this, please do. And in fact, I mean, it's something that's hasn't really been possible before, but now with all of us and everything like that there are no real excuses. So this was done, you can see, in the UK Biobank. This type of research wasn't really possible before UK Biobank scale data was made available. I'm not going to talk much about this. This talks about this is the example. So this slide, anyone can go back. This is the paper I published, right, Genevieve. So she shows basically the credible set. I was talking about this like credible set analysis when you have your GWAS and you've zoomed into a potentially causal region. In this kind of left panel, you have all European ancestry individuals, and in the right panel, you're mixing European ancestry with very diverse ancestry individuals, African as well as Hispanic and Latino. And you can see that the pattern of LD is actually kind of similar when you get from the total analysis, but because of the haplotypic structure, your credible set actually kind of bolsters one single gene that's tagged by all of the different haplotype structures across populations. It looks like we've got a couple of questions, or about one question. So I answered Akshay's, I did a no-no, I answered Akshay's question without actually hitting the button. And Akshay has another question. Akshay, this is great. Why don't we do a panel? You know, you can moderate the panel next time. Is the prediction accuracy being higher in European as a function of a greater availability of data, or are there other reasons as well, and is illness being more heterogeneous in one group as compared to other? It's pretty much only the former, the availability of data. So there are culturally unique factors that make studying things like psychiatric illness more difficult on the African continent and in African populations. There are certain aspects of culture that make studying Caribbean Hispanics very difficult in aging and in any kind of research that involves biomaterials. This is something we heard a bit about on day 2. Yeah, day 1 and day 2. There are challenges, but it's not studied, so we don't. It's like for people who live in those populations and work in those populations and study, deliver psychiatric care in Uganda, there's no human difference between the afflictions. There's no human difference in terms of the types of symptoms. The data does not exist to even make those comments. The data that do exist are genetic data that tell us that there's more diversity, it's more allelic diversity in Africans than in any other group or any individual of African ancestry than any other population on the planet. And that is only a very rich resource, because what it means is that you have a set of perturbations, natural perturbations at the population level that don't exist in any of the European samples, and any one of which could be causal for some type of disorder, or just, it doesn't even have to be a disorder, is associated with some phenotype. And because that variation doesn't exist in our populations, we can't even begin to measure it outside of a simulation or an animal model. So it's the former. There's no reason to believe that we can't get the level of prediction that we have in Europeans for any other population. What can go wrong? So talking about NSAIDs, anti-inflammatory drugs, highly prescribed, cardiovascular illness, there is quite a few drugs that have guidelines that are affected by genetic background. And cautionary tale is this drug called Plavix, which earlier this year there was a ruling. This was going on for quite a long time, for like 5 or 6 years. It was Sanofi and Bristol Myers who make this drug, and they had to pay the Hawaiian government almost $1 billion because they were accused of knowing about a body of research that I'll kind of outline for you in a second, that put the entire population of Hawaii at risk for adverse effects from this blood thinner Plavix, which is prescribed for post-operative care and prevention of blood clots following cardiovascular surgery. So its generic name is clopidogrel. And I brought up these CYP genes before, these liver enzymes, and it turns out that clopidogrel is processed by this CYP2C19 gene, the product of this CYP2C19, and there are alleles for this gene, the functional alleles that metabolize the drug and turn it into its active metabolite, which kind of does the job. It makes it effective. Right? It allows for the decreased platelet activation. And you take a look at the bottom. So the bottom pie charts show the frequencies of different alleles. Now here's something to keep in mind. Now we're going a little beyond just a single SNP. Alleles can refer to not just one SNP, but a haplotype, 2 SNPs, 3 SNPs, those blocks that I referred to before. It's a bit more complex. So this is why it's not just 1, 2, or 3 genotypes or 1 or 2 alleles. Individuals can be grouped according to this allele into one of 5 groups. And depending on the frequency of the genetic variants that make up this allele, you have different frequencies. And take a look at the pie chart for Asia. Individuals who have East Asian ancestry and also island ancestry, so this is a very unique population in the United States of Hawaii, have a much, much higher proportion of individuals with the yellow allele, very small proportion of people with that blue allele. Well, what does that mean? I mean, I thought I had a couple more slides on this. I guess I didn't want to delve in too deep, but I can summarize it briefly, which is, turns out that population of people who have this shift. So this particular allele basically is an allele that you need to have in order for, I shouldn't say need to have, I'm going to get confused on this. Someone can correct me here. It's the allele that, it might be the red allele. I have better slides for this. The point is, there's a much higher proportion of people in the Hawaiian population, because of their ancestry, their genetic background, that have an allele that changes their metabolic properties in a way that basically renders the drug useless. So you don't have a warning on the label that says, by the way, because you have, because we know from the demographic, the drug company should know this about its target population, because of ancestry in this population, because there's research looking into the allele frequencies in this population, there is a higher probability that you fall into a genetic category that makes this drug less efficacious. And that all has to do with this kind of precision medicine. And because the company did not actually put this warning on their label, so this goes back kind of a while. They didn't have this warning on the label. They were accused of knowingly putting their patients at grave risk of serious injury or death to increase their profits, basically by saying this company knew that just because of this genetic variant, something we can harness for precision medicine, there's a higher probability that it won't be effective for the population of Hawaii. But at the time, at least, the research was a little bit up in the air, and there are studies back from 2010 showing that there is no effect of this genetic variant on the efficacy of this drug to prevent kind of post-operative complications. But they were just doing it for their profits, kind of holding back this knowledge. So got to be kind of careful. They fixed it. They did end up fixing it. They added a boxed warning for Plavix. So it's not fun and games, but it's all having to do with diversity. It's a really kind of sound genetic mechanism that makes sense, and as soon as it's beyond just kind of saying we can't predict an illness theoretically. It might cost the company $1 billion. It might also have killed people, and certainly resulted in people taking a medication without knowing the full risks that were involved, thinking it was more efficacious than it actually was. It's a huge market for pharmacogenetics, and it's only getting bigger. About a third of the pharmacogenetics market is oncology, and it's biggest in North America. I guess that's kind of what I wanted to say, but it's really big money. And I mentioned a bit before that people aren't really willing to pay. Governments aren't really willing to pay unless you can show that it really kind of has an impact at the public health level, and I don't know if people really want to pay. And you can ask people about it, and you can ask insurance companies about it, how they feel about this. I mean, it's funny for insurance companies if they think pharmacogenetic testing is a worthwhile thing to do. And And so individuals who identify, if there's payers, providers, or just kind of societal perspective, individuals who felt that, this basically, this review is not a review of individuals, it's a review of studies, basically studies trying to determine this. Who believes, that this is a worthwhile investment? It turns out that both, payers for insurance plans as well as insurance providers believe that it's a very cost-effective thing to do. Pharmacogenetics will save money in the long term if you can predict the effectiveness of a treatment if you can predict someone's risk for disease and possibly avoid it, you'll avoid costs downstream. But individuals who are trying to look at the effect or studies that were trying to assess the effectiveness, the cost effectiveness of pharmacogenetics from a societal perspective, so saying is it good for society, or is it perceived well by society? Most actually said no, that it's not, and that's a strange kind of perspective issue. This idea of kind of genetically predicting your fate or genetically kind of tailoring medications to you, which is kind of counterintuitive and was a surprising result. And then, of course, part of it may be the lack of part of it's on us, I guess, the lack of really convincing data. So it doesn't necessarily surprise that people feel it might not be a good thing that we move into this space of saying you go to your doctor, and instead of what would be very traditional medicine, having a conversation about how you feel and your lifestyle and whatever else, and just kind of moving along, you get the drug and maybe it works or maybe it doesn't work. What if you're over-tested, over-diagnosed, over-treated, and overcharged at the end of the day, pushing patients into this kind of box of we need a blood sample and we need to put you through this assessment and everything else, all so that we can make some kind of prediction that so far none of the algorithms are doing very well. Would you subject yourself to a day of testing if the probability of of some integrative algorithm actually accurately predicting a treatment response for you was going to be 65%, like 15% over chance? I mean, maybe not. Algorithms and models today would claim better performance than that, but of course, in the generalizable sense, there isn't really much data there. So it's just something to think about, and a perspective that exists. And also from that societal perspective, there's risk, but there's legislation. So whenever people ask, about, are you afraid of having your data made available, right? I do 23andMe and put it online. And as I said, I've submitted mine to the TopMed imputation server because I want to have I want to be able to look at genetic variants that weren't directly on their panel. But both Canada and the USA, Canada was actually behind the boat on this, have passed these acts that prevent anybody, any employers, insurance companies, etc., from requiring people to undergo genetic testing or disclose genetic test results or base any kind of any kind of coverage or employment status based on genetics themselves. Although the USA Act, which is older, you can say here, it does not protect against genetic discrimination for life, disability, and long-term care insurance, and neither for small businesses, military, healthcare providers, or other groups subject to exceptions. There are exceptions. So it's kind of strange. And yeah, he's kind of buried in there. So, I mean, nice to be in Canada for that. So finally, utility and implementation. We talk about clinical utility. What it means, this is simple. I'm not going to go too much into this. It's just about the value of information to the person being tested. It's not just about predictive accuracy. Clinical utility for something could be something that gives someone peace of mind. It could be something that reduces their wait time. It could be something that makes them feel more informed about what's going on. It's not about curing an illness necessarily. So again, even if no interventions are available to treat or prevent, there may be benefits associated with knowledge of a result. if you're thinking about utility, and I love that Abi brought this up, most research is not useful. That's the case. It's certainly the case. He came at it from a perspective of reproducibility and transparency is really important. Another angle is the actual question, right, that's being asked, and is the value of what you're delivering actually of value? Does it have a utility? Can it be used for anything? And these are some kind of outline of questions, basically, that you can ask yourself, and that any research program or funder would kind of ask of what you're doing, to understand if the research is useful from a kind of clinical utility perspective, right? Is the health problem big and important enough to fix? It seems self-evident, but sometimes research can be done on questions that aren't big or important enough to fix. And we can argue about what that actually means, and it's all about kind of due diligence in a large kind of way. Has prior evidence been systematically informed? Is the study large enough, right, statistical power? Does it reflect real life? Does it reflect patient priorities? Is it worth the money? Can the research be done? These things in reviews are found to be enormously underdeveloped, not clear. If you're ever trying to write a very convincing grant document or award application, think about these questions and address them explicitly, as people will be looking for the answers. And this is kind of reviewing these aspects of what a kind of useful result means and how often they are actually met in literature. So as you can see, not often. They looked at major medical journals and then kind of all clinical research. And, at least in the major medical journals, a majority of studies address information gain, right? What stands to be gained, but it's actually rarely addressed in all clinical research. Is it patient-centered, very rare, uncommon, right? Value for money, unknown, rare assessments. It's like things that we think are there that we might impute in our reading of these papers are often missing. One proposal, kind of a way that is a general framework for kind of developing useful, informative, patient-centered, and integrative precision medicine is through, clinical trials. Basically, you cannot assess the efficacy. There's no standard for evidence of addressing the clinical usefulness of an intervention or new information in guiding treatment without performing a clinical trial, without performing some implementation of what you're doing. And so there are guidelines that have been provided for designing such a study, and they mostly center around, again, you see this little smartphone of someone clicking, data being collected throughout the course of a day, right? Real-time measurements of behavior and digital phenotyping, a huge part of that, as I mentioned. So it's really important to think about the principles that Abhi was talking about. This is a specific example of using EEG to possibly guide treatment with transcranial magnetic stimulation, and kind of ways to adapt the treatment over the course of the program depending on the measurements that were made in the first place. And so, if you're thinking about ultimate clinical utility, it's very useful to kind of think back to where that evidence is coming from, and if it is a diagnosis in a population, or if it's a case control discrimination that you're looking at in a case control data set, even if it is a truly clinical population, without the intervention strategy and the correct clinical trial design, you can't really assess the magnitude and validity of that intervention, and that centers around, even after that scientific evidence is in place and even after you've established some specificity, you need to perform implementation science, and this is something that is virtually never done. This kind of set of guidelines, there's a framework that was published in 2009 for what useful and kind of meaningful implementation research would be. Basically, the idea is, if I come up with a map of the human brain that I can apply to anyone's fMRI scan and say, okay, if you have this pattern, or if you fit it, or if you show this degree of activation in these two regions or between these two regions, you're 80% more likely to develop a psychotic episode or to experience a psychotic episode over the next 5 months, right? That's all well and good, right? And we heard a bit about this from Joanna earlier today. And she will welcome any questions that you have based on this, because she and the Brain Health Data Bank group are quite involved in this process. How do I take that? Let's say that gets published in Nature or something. I mean, like, let's just say that we're the greatest scientific discovery ever. You're still at the very beginning. You're really at the very beginning of this, because what do you do, right? And you've got to think really practically, really pragmatically. What do you actually do? So I'm a scientist. I'm on the 12th floor of the CAMH building. Do I go and wait in the lobby of the emergency room and find a patient as they're coming in and kind of listen to their, the triage nurse and try to figure out, okay, maybe they fit the inclusion criteria, and then hang out around like the MRI scanner and try to like sweet talk the technician into getting their scan, and then go and process it on my computer, and then come up with a prediction, and then just like spam all the doctors in the hospital and say, who has a patient that looks like this, right? It's a bit odd, but these are things that matter. How do you tell the doctors in your community, in your hospital, about this research? How do you tell them it's worth listening to? How do you get people's scans? How do you get the scans out of the clinical, highly protected, firewall environment of clinical scanning into a research setting? How do you demonstrate that that's going to be useful? What type of study do you do to say, I can get doctors to listen to what I have to say? I can give them information that they understand. When they understand this, they can communicate it effectively to the patient. When they communicate it to the patient's receptive. When the patient's receptive, the doctor makes a decision, and that decision ultimately goes back to benefit the patient in some way. Like, all of these steps are not thought about in implementation science. This is a review of studies in psychiatry, of examples of models that have been developed for, in a lot of cases, in almost every case, the vast majority are actually prediction models for the emergence of psychosis. So it's this idea that you can have outpatients who are diagnosed with a variety of disorders. They can be it can be major depression, it can be bipolar, it can be schizophrenia. But having these diagnoses put you at risk for potentially, more than others, of having a psychotic episode, which could be a very damaging and very severe experience. And these models, there were 50,000 of these studies that they screened. They screened for eligibility. It's not useful to me to have this top bit, actually, because they looked at about 1,000 articles that they read through. They found 584 predictive models. Now, not all of them are for the emergence of psychosis. A lot of them are. But these are just generally precision psychiatry models for response, treatment response, everything else. Of these 584 models, 89 of them were validated. So this is just internal and external validation. You see 10% were internally validated, 4.6 had been externally validated. It's tested in an independent data set, and 0.2%, one study of the initial 51,000 screened and 500 models had actually done science in their part of their study to understand how they can implement the results of their study inside their care setting in their hospital. This is recent. This is like 2 months ago, this study. This is not like, oh, now there are examples. This is where we're at. This is the study, by the way. And this in this particular case, it's transdiagnostic. They're not looking specifically. It's like at individuals who are diagnosed with schizophrenia. It's just in a real-world psychiatric care setting. There's a couple of different hospitals. They are just looking to understand who is at risk for psychosis across diagnoses, across the whole pool. And this is the result. So again, this is the one study that did an implementation assessment for their model. They, in doing so, basically had to screen patients as they came into the hospital, apply their algorithm, right? It's just that. So you enter all these different data points, and we'll talk about these models more tomorrow. They enter their data points, they get some prediction of, almost 4,000 patients that came through. They flagged 117 that were at risk for psychosis. So remember to think back to like the polygenic risk stratification. Those analyses are looking at like the top 10%, people in the top decile of the score. This algorithm is is trying to understand, how many people, again, this implementation, practical, how many people is the algorithm going to light up for, right? You can't light up for someone if they have like a 0.0001% increased risk, right? That might have more adverse effects than not. So the algorithm lit up for 117 people. 2 of them had no contact information. 115 of them were identified. They had a framework to get in touch with clinicians. There's a whole series of, there's one email and then a follow-up email and then a third email and then a phone call, and if nothing, then that's it. Anyway, by the end of the day, 39 patients out of 3,700 were referred, kind of basically changed off of their care as usual course for specific treatment having to do with their elevated risk for psychosis in this. So, success, right? Is that precision medicine? Well, it is. You be the judge. 39 subjects, and so there are a lot of ways in which this implementation can fail. And Joanna is the expert on this, and she's been kind of studying this. People don't share the same agenda, research purposes. You might have clinicians or people, stakeholders, patients, families of patients who think that your risk prediction is nonsense. We don't want to do the same thing. We want to attach something else. Language, really developing those dashboards that Joanna showed. That's not just something that we came up with. That's not something that she came up with. It's not something that any scientists just came up with. It's a result of study. It's a result of working with clinicians and working with people who will actually be looking at this information, because our language might not be in common with them. It might not stand the test of time. We might not publish implementation for our studies because we have a short-term mentality in terms of our studies. Why do that implementation work if we just want to kind of publish work and get to our grants? Doing implementation science and dedicating time to it and understanding how to deploy your work it takes time, and it's hard, and it's just really not done. And I'd say, aside from the scientific elements and the lack of available data, the reason why, probably the biggest reason why we don't see precision psychiatry in practice is because of the lack of implementation scientists. Second only to the fact that most of the predictive algorithms that exist are insufficient or lack external validity. So, anyway, I'll end with this little pretty picture here, which kind of sums up the research ethos, kind of design of what my group is part of. It kind of summarizes a lot of what KCNI is about. But we use external data sets, bring them together, model external phenomena, but also take collected data from inside the hospital. You see, we integrate directly with clinical services. This slide would be paired best with Joanna's slide on Brain Health Data Bank. Basically, the intersection of bringing together and modeling these huge external data sets where we have numbers and statistical power with collected data from inside the hospital, and KCNI is uniquely positioned there. We can access clinical data from a number of care pathways and generate these predictions that feed right back into the clinical dashboard. And we talk to the clinicians. We interact with them directly, and that's part of this implementation science platform. So I'm going to pause right there. It's been an hour and 15 minutes roughly. Doesn't look like I've kind of been addressing some questions as they go. Actually, I had a point. I think there's a challenge of incentives of knowledge translation in both the publishing side, where a reproduced result is unlikely to be published in Nature, and also changing the incentive systems in the clinical side that those features are adopted. I wonder what would value-based care look like in the case of mental health. Yeah, there's a lot to unpack there. It's a good thought. Incentives. A lot of it is incentives for sure. For knowledge translation, for publishing, it's complicated. So I don't have the answer there, and I'm not a philosopher. Well, we all like to think we're philosophers, but I'd rather not just kind of go on about it. I'm trying to give you the perspective of where we are, how to think about it. I think many of us are capable of recognizing recognizing just the fundamental nature of those problems, but the important thing to do is to turn around and come out of a summer school like this and go back to your lab and go back to your project and just have those wait a minute moments where you do kind of think for a second, okay, this is all well and good, and I have my own motivations, right, and I have to finish my degree or I have to do this or that, but is there something I can do? Is there a next step here? It doesn't have to be a whole new modeling. It doesn't have to be another paper or a whole kind of expedition or anything. Is there something else I can do to make this result more externally valid, internally valid, or address explicitly in the writing of this paper or in the writing of the abstract or the delivery of my presentation some of these aspects of utility that we kind of take for granted in our work? Some of us don't take it for granted. I know, it can be an endless source of disillusionment to be working very hard on something that you fail to see the utility in. So think about these things and know that there are recommendations out there. There are ways, specific questions to ask, and ways in which you can kind of couch your effort in this pipeline of utility. So anyway, that's it for me, really. I have this section on introduction to integrative models, which I was planning to kind of go through really quickly. It's only a few slides. I don't think I'm going to do that. I think I'm going to save it for tomorrow and actually spend a bit more time on some model statistical stuff tomorrow. It's been a long day. Has it been a long day? It's been a long year. It's always a long day when you're sitting in front of a computer. We can do more jumping up tomorrow morning. We'll get back into the mood. Science is exciting, and we should all be, I know Aaron says there's always time for more science, and I know there's always time for more science. So if I know the crowd is, all so silent, we could do a little bit more, but I'm not gonna poll it. I think we'll hold it there for the day. So in a couple of last minutes, is there anybody who has a burning question? Thanks, Sunny. Time does fly when you're having fun. Yeah, you should have fun. I like to think of it this way, Sunny. In the science and in this kind of research, it's pretty heavy, right? It's really important. You're thinking about all the suffering that happens and how you kind of change lives. You can take the work very seriously, but don't take yourself too seriously. I think as long as you do those things, you're going to be okay. Because if you take it all too seriously, then you're really, you're going to miss out, and you're going to drive yourself into a wall. But time does fly when you're having fun. So we're going to have tons of fun tomorrow. We're going to bring everyone back in to talk about all aspects of this. I'll be back to talk about digital health. We'll have Joanna as well available to talk about implementation, about examples of implementation. Bring your questions. John, Andrea, Shreejoy, Itai, Laura, Dan. I think we're going to have Victor and Brett as well. The whole crew. So bring your questions. And we'll get at that in the afternoon. The morning will be integrative modeling, examples of integrative research, and thank you all for attending and bearing with me in my time of need with technical difficulties on the microphone. So if there are no other questions, I will thank the primary question asker, Akshay Mohan, and all the commentators for their attention and participation. Aaron's posted the GatherTown link. I'll pop over to GatherTown briefly to kind of catch up with anyone who has remaining questions. And yes, the post-workshop survey. The post-workshop survey is really, really important. It's really important to everybody. Anyone who teaches, myself, we have a lab, a research lab, a lot of how I am seen in the eyes of the institution and the powers that be is through feedback from forums like this and for courses, and we put a fair amount of effort into it, and your feedback would be greatly appreciated, whether it be good or bad. If you hated it, tell us you hated it. If you love it, tell us you love it. And and be easy on the technical difficulties, because just it happens. So thanks everybody. We really appreciate your time, and we'll see you bright and early tomorrow morning.